the inaugural Dorothy Winfield Phillips Chair. She received her BS in chemistry with a concentration in business from the University of Louisville and her Ph.D. in analytical chemistry from Indiana University. Under the mentorship of Dr. David Klamer, she developed proteomics methods to study aging in fruit flies. She continued her aging research as a Lyman T. Johnson postdoctoral fellow and a professor with Professor D. Allen Butterfield at the University of Kentucky. During this fellowship, she began to focus on neurodegenerative diseases and disorders such as Alzheimer's. She received the UNCF Merck postdoctoral fellowship, and in 2009, she accepted a position as the assistant professor of chemistry at the University of Pittsburgh. In 2017, she moved to Vanderbilt University as an associate professor of chemistry in the Dorothy J. Winfield Phillips Chair. She is currently a professor of chemistry and a principal investigator in the RASR Lab. She's the faculty head of the Murray House and holds a secondary appointment in the Department of Neurology. Additionally, she is a faculty member in the Vanderbilt Memory and Alzheimer's Center, as well as the Vanderbilt Brain Institute. Dr. Robinson is the most immediate past president of NOVASHE, the National Organization for the Professional Advancement of Black Chemists and Chemical Engineers, and she's a faculty co-advisor for the Nashville chapter. Let's welcome Dr. Robinson.
Thank you all for coming today, and thank you, Dr. Quarles, for the invitation to come and share. I'm really excited to share with you some of the work that's taking place in my laboratory at Vandy. And I apologize to you and also to my students who are doing excellent work, but that I won't have time to talk about that cover like different areas of the work we do in the lab. I do want to acknowledge the team that we have in the lab. So without them, none of what I'm going to tell you about would be possible. We've had some recent graduates from the lab in the last semester. So some of these folks have moved on to postdocs and graduate programs and other positions, but I'm really grateful for them and also for the funding that has supported the work that we do. Okay, so I have 5 goals for our time today, and they're shown here. So the first thing that I want to do is teach you a bit about disparities in Alzheimer's disease and convince you that proteomics is a tool that can be used to further our understanding of these disparities. You may or may not be familiar with proteomics measurements, so I do want to teach a little bit about what that means and how we do those measurements. And then I want to give 2 examples of how we're applying this. So one that is to further disease understanding and pathogenesis, looking at post-mortem tissue, and another is working towards biomarker discovery efforts in plasma tissue. And then at the end, I'll just give you an overview of like the kinds of other larger ongoing projects that are taking place in our lab now and that we hope to continue in the future. Okay, so let's start in the beginning. So prior to COVID, this would be a survey of what looked at the top reasons why older adults would die. And so you see at the top would be heart disease, cancer, and then if you keep scrolling down the list, you'll see that Alzheimer's disease is number 6. And it turns out that medical advances have gotten really good. And so the number of individuals that are dying from things like heart disease and cancer is actually going down. However, because our aging population is growing tremendously and we don't have cures for Alzheimer's disease, the number of deaths that are being attributed to Alzheimer's disease has been increasing dramatically. It turns out that Alzheimer's disease also disproportionately affects different parts of our population in the US. And so here's data that looks at the prevalence of Alzheimer's disease in African American and non-Hispanic White communities. And what you can see is that African Americans are 2 to 3 times more likely to have Alzheimer's disease than non-Hispanic whites. And in Hispanic Latino communities, that number would be like 1.5 to 2 times. So there's disparities in incidence and prevalence. And then there's also disparities that have to do with the financial burden of the disease. So although African Americans make up less than 14% of the US population, they carry about 1/3 of the total costs that are related to Alzheimer's disease and other dementias. And most of these are costs that have to do with like caregiving, giving up one's job, bringing somebody in the home to care for a loved one. And it's pretty tremendous in terms of the actual amount of our economy that is tied up in Alzheimer's disease care. The other thing that's really interesting when you start looking at minoritized communities in the US population is what their perceptions are about medical research and healthcare. And so what this data shows you from the Alzheimer's Association is that a number of US adults from racial and ethnic groups report that they have experienced or often experienced racial discrimination when they're seeking medical care. And then also some of these individuals believe that medical research is biased against people of color. And I won't give any personal anecdotes now, but I'm happy to talk about a personal one that just happened 2 weeks ago at the dentist's office, if you're interested to know what that was like. Okay. But the reason why I wanted to share that is because this is kind of the context that I was getting into when I started this part of my research program. And what I was learning is that if people believe that medical research is biased against them and also basic science research, then it's important to understand what are all these barriers that are preventing them from also wanting to participate in research. And so mistrust in medical professionals, researchers, scientists is a really big deal. And there's a lot of long-standing, history of mistrust in the US community as a whole. And then there are all these other reasons why people may not want to participate or may not have access or ability to participate. And so I actually was quite interested in this because at first I just wanted to like analyze samples as a chemist, is what I do. But I recognized that this was a bigger problem than just the chemistry that I wanted to conduct on biological samples. And so I got into some community engagement and outreach work when I was at University of Pittsburgh, and I was a part of the Alzheimer's Disease Research Center. And these are some of the co-investigators that worked with Dr. Jennifer Lingler, who was in the School of Nursing. And what we did was we started a project to understand what were the reasons why people from this community did participate in research. And could we learn anything from those narratives and use them to help encourage others to participate in research? And so what we did was we conducted interviews with either caregivers or the individuals that were in the Alzheimer's Center study directly. And then we developed materials from those interviews. So what you see are two examples of like brochures that might show up in a doctor's office, a community event. We turn these into videos that would play at community events, and also that showed up on social media, and we had a really large social media campaign. So we would learn a lot of things from the community about what they learned about disease, how they felt about research after viewing or reading the materials, and they would be very transparent in what they, said. So, I like the statement here. It's very clear what the study is going to be about, but the font's too small. Or, I get that this is about Alzheimer's, but this guy doesn't look like he has Alzheimer's. He looks too good looking. So maybe, we actually see somebody who actually looks like they have disease. So this is a lot of like qualitative research, which is very different than the analytical stuff that I do. But it's actually really insightful and helped us to build up pathways to recruit more people into research studies. And so I started at the University of Pittsburgh, and then we got some funding to continue the work at Nashville, which there we were interested in doing similar types of focus groups, but also trying to understand if this community, which is closer to the South, closer to Alabama and some of the Tuskegee University syphilis studies, if they have differences in how they perceive research and their interest to participate. So this is some ongoing work, and we've published on some of this, and that will be forthcoming. Okay, so the other piece of what we've learned in talking with community members and also in doing this work is that it's not actually really straightforward to just put people in the groups in the way that I'm going to describe for most of our research, without recognizing that each participant is an individual that has a lot of lived experiences. So everything from like what they eat, what they have access to eat, what kind of stressors they're exposed to on a daily basis, what their education and quality looks like, their health, other conditions, all of these things are really important for us to understand and how they contribute to the biological signals that I'm going to be describing today. So I want to acknowledge this and just disclose that in the studies I'm going to tell you about, we weren't able to capture all of those different elements because we're looking at samples that go way back to like 80s when a lot of this thinking had not been brought to the field yet. So I mentioned that comorbidities were important in terms of understanding someone's experience, in particular for Alzheimer's disease. If you have any of these comorbidities like high blood pressure, hypertension, these are things that would increase anyone's risk of developing Alzheimer's disease. And in the African American population, there's a high incidence of cardiovascular disease and things like hypertension that can contribute directly to some of the pathological features of this disease. There also are things like genetic components. And so maybe you've heard of ApoE being one of the genetic risk factors for Alzheimer's disease. So that is true. However, like the results are really mixed and controversial in African American Black populations. And so in some cases, they can be predictive, in other cases they're not. And so there are a number of other genes that have been shown to have better effect sizes at determining a risk of Alzheimer's disease in this population. And so we're doing some work looking at APCA7. And so when you take, comorbidities, genetic factors, and all of those other socioeconomic factors that I just described together, all of these things are contributing to the pathological features that show up in Alzheimer's disease and increased risk and prevalence in certain populations. So if you look at a brain that doesn't have Alzheimer's disease, it will look something like the one shown on top, very healthy and vibrant. And when you start to look at someone that has advanced Alzheimer's disease, you notice that the size of the brain has shrunk and that there are a number of deposits that are forming on the brain. And so these deposits come from amyloid beta peptides that aggregate and make senile plaques. And then they also come from the hyperphosphorylation of a protein called tau that results in these tangles within neuronal cells. And so one of the questions that I was curious about was whether or not these basic pathological hallmarks manifested in the same way in African American communities compared to non-Hispanic white. And so there was a study that a researcher at Rush University Medical Center conducted, where she had been following African Americans in aging over about 20 to 30 years. And what she noticed was that in some of her participants, that the African American individuals, when you looked at their brains, not only had pathology of Alzheimer's disease, but also were having higher incidence of other kinds of dementia. So it turns out that this depended on like where exactly the participants were recruited from, so whether or not they were coming from the community or whether or not they were coming from clinics. But this was some piece of data that I ran across that was showing that maybe there's more complications that we need to understand between these groups. The other piece of data has to do with looking at biomarkers for Alzheimer's disease. So this is looking in cerebral spinal fluid at that protein I just described that makes tangles, total tau. And what you see here is that across different ages of adulthood, the levels of tau are lower in African Americans relative to the non-Hispanic whites in this study. So this is a study that's been done a number of times, not so many, but I think in the small number that it has been done in, what this really kind of lends sight to is the fact that if you have a part of the population whose baseline for a particular biomarker is low, but you don't know that, then you would have parts of the population that would go undiagnosed for having something if this would be the baseline level at which you're going to develop a biomarker. So I think it's important to make sure that these studies are broadly inclusive so that we can understand where these biomarker levels need to be set, whether it's coming from CSF or any kind of other fluid. Okay, so I wanted to kind of get started with this work, recognizing that there would be barriers of being able to recruit people into studies, but also being able to have access to tissues. And so I started by looking within the field of proteomics to see what were some of the major studies that had informed what we knew about Alzheimer's disease, and what did the diversity of those cohorts look like. And so it was fairly low in most cases. And so I decided to try to conduct a pilot study that would allow us to start to increase the number of African American adults that were represented in the proteomics literature. So I was at the University of Pittsburgh, and they are one of about 35 centers nationally that recruit participants in Alzheimer's disease research and have biobanks. And so I started with Pittsburgh. They had about 1,200 samples altogether. And some of these are like following a person multiple times over many years. And what I was shocked by was the fact that out of 1,200 brain tissues that they had, only 6 to 10 of them came from African American participants. And so that is another reason that drove my storytelling work. There were more samples when we started to look at blood draws and plasma. So there were about 130 that were there. So we requested some funding from the center to do some pilot work, just starting with everything that was going to be available in the center. And then we knew we had more non-Hispanic white participants to randomly select and match from. And so that's kind of where this work started. So I'm going to take an aside from this, but this is a backdrop of where we are in terms of disparities being a problem that I think that proteomics can be useful in solving. So now let me teach you a little bit about proteomics, and then I'll come back and tell you about what we learned from the first pilot study. Okay, so proteomics is essentially the study of all or some subset of proteins that are present in a given tissue that you may be interested in analyzing. This could be a cell, it could be plasma, it could be brain tissue, brain slice, it could be a whole organism, depending on what you're interested in. And what we're going to do here is extract the proteins from our sample. And then in order to make them more feasible to analyze using mass spectrometry, we're going to chop them into pieces using chemical enzymes. So typically we use trypsin or lysine, which cleave at every lysine or arginine residue. So we make our mixture more complex by doing this. And then we have some tricks that we play using some chemistries to multiplex that I'm going to tell you about in a minute. And then because this is so complex, we're going to take advantage of different levels of chromatography in order to separate the peptides out before we introduce them into the mass spectrometer. And so by the time we finish with our analyses from the mass spectrometer, we use a lot of database searching that tells us what was present in our sample to begin with, and kind of you can just keep doing this process again and again until you get through all of your samples. So my group is a mass spec group, and we rely heavily on mass spectrometry technique as our primary detection and analytical tool. And we get 3 pieces of critical information from the mass spec. So I'm making this really simplified, but it's not this straightforward. So there's a lot that goes into getting a sample, a lot to actually running in a mass spec. But the first readout we get is a high resolution mass to charge value that gives us an accurate mass of a particular peptide peak. The second thing that we do is we can isolate different peptide peaks, and we can subject them to fragmentation with high energetic collisions, and then we break them into pieces. We measure the masses of those pieces, and we can look at the differences between peaks and figure out what the amino acid sequence is for that particular peptide peak. And so because we have so many of these spectra that we generate in an experiment, we use a lot of software algorithms to do this part of the work for us. And then the third piece of information that we get is where we do the quantitation. And so we have some signals that show up at low mass discharge in our mass spectrometer that we're going to use the intensities or the areas under the peak in order to compare how each protein changes between the different groups that we're interested in. That could be controlled disease within racial groups, genders, kind of anything that you're interested in. So to help speed up the process, we introduce multiplexing. So if we just go through that whole circle that I described, that might take maybe 5 to 6 days, depending on how many samples to go from beginning to end. If you have like 40, 100 samples, then it would take months to do that process. And so what we do is we rely on chemical barcoding to help us put more samples into the mass spec at a single time. And so this is an example of a commercial tag that we use called a tandem mass tag, and it has 3 main parts. This part here, the N-hydroxysuccinimide ester, is the part that reacts with the lysine residues on our peptide and N-terminal amines. We have a mass balance region and a reporter ion region, which can vary depending on the reagent. And so the idea here is we can use heavy isotopes like carbon, 13, deuterium, oxygen, nitrogen, and we can move where those heavy isotopes are between the balancer and the reporter ion region so that all 6 versions of this tag have the exact same mass, but the location of those heavy isotopes are different. And so now if we take 6 different patient samples and we react the peptides from those samples with one of these 6 different tags, we can mix them together and we run them in the mass spec and get the first signal. We only see 1 peptide peak because all of the samples, the peptide shows up at the same place. Once we do the fragmentation experiment, we actually break this bond here on the tandem mass tag. And so this reporter ion gets released, and we measure that at low mass to charge values. And so the concentration of this reporter ion is related to the concentration of the peptide, protein in the sample, and then that's where we get our quantitative information. So at the time we started, you could buy these kits where you could do like 6 samples at a time. Now we can do 18 samples at a time. And in a whole other part of my lab where we do analytical method development, we actually have technology to double and triple how many samples we can put in the mass spec at a single time. So one way we speed up our throughput is with chemical multiplexing. The other approach is to do liquid automation. And so we take our manual pipetting tips, and we have a team that worked really hard to transfer those manual pipetting tips to a robotic liquid handler. And so this liquid handler has a bunch of 96 well plates that can be used to place samples, place sample reagents, and run reactions. And so it can just go in and pick up samples. It can move them to another plate or a SPE device, depending on what your protocol requires, and then it can run those reactions. We also have a second arm that can run larger volumes. So this arm has 8 pods that can do about 5 milliliters. So we have to figure out how much volume that we need ahead of time. But in doing this, it allows us to be able to work with the number of samples at a single time, prepare them, and then be able to reduce our total sample prep time. So ideally, we can run up to about 12 plates at a time on this device, which would allow us in the future to get to 1,150 samples that we can run simultaneously. But it makes me really nervous to think about working with that many samples, which defeats the purpose of having this. However, the most we've done so far is about 4. And so, some learning lessons along the way. So as we gear up into work on being able to handle a larger number of samples that we're going to take from the sample prep all the way through to the mass spec, one of the things that we realized we needed to implement in the lab was robust quality control measures. And so by that, I mean, how do we ensure that every sample has been prepared in the same way, our reactions are efficient, so that at the end of the day, when we look for differences, we know that they're due to disease and not because we missed reagents or some user had a misstep. So Nakesa had worked in industry as a master's with a master's degree for a couple of years, and so she helped us to spearhead this part of the work in the lab, and these are some of the technicians that helped her. And so essentially every part of our sample prep process, we stopped and took the time to figure out what would be a good quality control sample that we could embed into our 96 well plates or into our process, that we could monitor its performance to make sure that part of the process was going well. So we have processes with quality control for the sample preparation. And then Dr. Kyrie Patterson, a postdoc in the lab, developed protocols for the LC mass spec collection. And so essentially, he has different metrics. So he has about 8. I'm showing you one example of one, where we can look at how well a quality control sample performs for this given metric over the course of time or so many injections. And what happens is once you start running these samples and you have months of data and you have 100-some quality controls, you actually have a really good idea of how many proteins you should see, what the level should be, how efficient the fragmentation should be on the instrument, all these different measures. And so anytime that you have a sample that is outside of where it should be, then it actually informs the operator to stop. And so now the student or the postdoc, they stop, and then because we have so many metrics, they actually know where they can troubleshoot in order to get the experiment back on track. So that might mean that they actually need to stop and do something with the liquid chromatography separation. And so they have an idea of what they need to do there, or it's the mass spec that's causing the issue. So prior to this, we were just kind of blindly like, well, go check this, go check that, go check this, go check that. And we have a lot of downtime, but we try to speed up the process. And so this quality control has really helped with the speed, but also the rigor of the work that we're doing. Okay, so generally that's about how we're doing proteomics in the lab. And the data that I'm going to show you has been the progression of going from manual sample prep workup to incorporating some of the automated steps. All right, so I said earlier that we took samples that we could get our hands on from the University of Pittsburgh, and that's kind of where we started. So this was work that Dr. Caitlin Sepler led. She's now at St. Mary's as a teaching faculty there. And what she did was we requested multiple brain regions from the African-American participants. And then we went and matched the non-Hispanic white participants to their characteristics based on age, sex, ApoE status. And the regions that we chose were regions that have known pathology of Alzheimer's disease with the plaques and the tangles that I shared earlier, but also that relate to brain functions like memory and executive reasoning and things like that. We had one brain region that we also ended up collecting called the globus pallidus that didn't have any known pathological hallmarks and wasn't really implicated in disease. And so that was, for us, we thought, a negative control, just to make sure our approach was not going to pick up on many changes there. And all of the work that we do is in collaboration with biostatistical teams. And so we have one led by Dr. Tim Holman at the Vanderbilt Memory and Alzheimer's Center, who helps on a lot of these projects. So we have 3 different brain regions that come from, in this study, I think was about 20 different participants. Each brain region gets processed through the proteomics protocol that I described. And then at the end of that, in most of the brain regions, we see close to about 2,000 proteins. And so some of the proteins only show up in one of the brain regions, depending on where it's from. But then what we're really interested in are looking at how these proteins are changing in people that have Alzheimer's disease and in the individuals that did not, but were age matched. So that's illustrated here in this Venn diagram. So the hippocampus, which is related to memory, you can see had the most changes in Alzheimer's disease in everybody in the study. And then the globus pallidus, which we predicted didn't have as many changes. So the volcano plots give us an idea of by how much those proteins are changing. So here on this axis is the ratio of the protein abundance from individuals that have Alzheimer's compared to those who don't, and it's a log 2 scale. And then the y-axis is the p-value, so how significant is that change? So everything in red is something that is significantly different, and if it's on the right, it means it was higher in expression in people with Alzheimer's. On the left would be lower. And so we can march down through different brain regions and find which proteins are different, and then we can also see what their abundance levels are. And again, based on what happens in each of those brain regions, then the number of proteins that we see changes accordingly. The other way that we can visualize how these proteins are changing is using a heat map. So this is a lot of data to kind of think about handling. And so what the heat map does is it clusters together proteins based on how they change in different people. So what you see here is that most of the people that are have normal cognition are on the left, and the people with Alzheimer's are on the right. And so most of those people and their proteins are clustering together. We have one large group of proteins that generally are lower in people that have Alzheimer's disease, their expression is, and then a smaller group of proteins that is the opposite. The thing that I really like about this particular way to visualize the data and just proteomics in general is that it gives us an unbiased way to actually look at the diagnoses that doctors make, and to maybe use it as a tool to determine if they misdiagnose or if they haven't seen how complex disease are. And so I say that because we were able to see some individuals that had a known diagnosis of Alzheimer's disease. However, their proteome looked more like people that didn't have Alzheimer's disease, and vice versa. So the more that we run these experiments, we actually see that the disease is very complex, and there's like a spectrum to what the profiles look like. Okay, so we can quantitatively look at proteins that change and know what levels they are, but then we can go further and try to understand overall what biological pathways are changing in these data. And so we can do that in each brain region. So here in the hippocampal region, we saw a lot of changes in mitochondrial dysfunction, oxidative phosphorylation, synaptogenesis signaling, which are all pathways that have previously been implicated in Alzheimer's disease. So that was encouraging to see. And then in the IPO, we can follow, for example, pathways that are changing, like glycolytic pathways. So this one is really cool to see because in PET images of Alzheimer's patients and age-matched normal adult patients, you can notice differences in glycolytic levels. So here you can see that glucose metabolism is a lot less in Alzheimer's patients compared to those that are age-matched. And the proteome results mirror that kind of result. So when we start looking at proteins that are involved in these pathways, we see that their expression is also lower, which means that they would be producing less glucose as well. Okay, but the real question that we had with this whole project was whether or not we were seeing differences based on race and ethnicity in the participants that we ran. So to answer that question, we conducted a statistical interaction analysis that looked at race and diagnosis based on each protein abundance. And what we learned was that there are proteins that behave in many different ways. So some proteins like this alpha-2 macroglobulin, you can see that the levels of it don't change in people that have Alzheimer's disease and are non-Hispanic white participants, but the levels are lower in the African-American participants that had Alzheimer's disease compared to that group of cognitively normals. There are other proteins that behave the same way in both groups of participants. And then there are other proteins that potentially have the opposite direction of change. And so these are like pilot data. The sample sizes were low, but this was sort of the first indication that we really needed to make sure we were having better representation so we could fully understand disease pathogenesis in this disorder. So we went back to our methodology, and this is where we started to bring in that automation tool that I described. We started to bring on tons of chromatography separations so that we can increase the depth of how many proteins we could see, but also make the process way more robust. So Jasmine in the lab spent some time doing this, and we were lucky to also get our hands on more samples from the Rush University cohort. So here we double the number of samples that we had. We also had multiple brain regions. But here what you can see is that we now can see close to 10,000 proteins by improving our analytical performance. And so Jasmine, with a different cohort of patients, did similar types of experiments, and she was seeing very similar results. So many similar biological pathways were changing in people that had Alzheimer's disease. And when we did the race by diagnosis interaction analyses, she also found groups of proteins that also had different abundances depending on what particular race and ethnicity the participants were coming from. So this is an ongoing work that we're doing in the lab, and we're really working to build up the number of post-mortem tissues that we have access to so that we can fully, one, replicate everything that we're finding, and then start to dig in and understand why some of these proteins may be changing in one group versus another. So that's one area of work. Everybody doing okay? All right, so let's keep going. So now let's look at blood and how we can think about biomarker discovery efforts. So everything I just told you is in postmortem tissue, so we cannot help those individuals. It just helps us to understand disease. In actual patients, the ways that Alzheimer's disease is diagnosed is based on like a clinical diagnosis where there's neurocognitive tests, there's family history, all kinds of things that doctors do. And then they also can get imaging. So they can do PET imaging to look for amyloid beta or tau tracers. And then they also can look for concentrations of amyloid beta peptides and those tau proteins that are basically coming from cerebral spinal fluid. So in order to get to cerebral spinal fluid, you are asking a patient to agree to do a lumbar puncture, which means they have a large needle going into their spine so that you can make that measurement. So one, there are some communities that do not want to agree to participate to do the lumbar puncture, thus you wouldn't be able to make those measurements. And there are also communities that don't have access to be able to get to the technologies where they can do PET scans or MRI scans as well. So you really need a way that you can diagnose disease that's going to be more accessible and somewhat less invasive than doing the lumbar puncture. And so plasma is a really good option for doing that because one, is something that most patients are familiar and agree to do. Most of us agree to do a blood draw when we go to the doctor, so we're not asking for anything that they normally don't agree to. But then also plasma has a lot of rich information about proteins that are secreted from brain and other organs because it's constantly involved in circulation. All right. So the challenge of working with plasma, though, is that it has a lot of analytical noise. So I got it. Yeah. Every room looks the same, believe it or not. There's always a panel right there. So the challenge of working with plasma is that it's highly heterogeneous. So a student of mine went and surveyed the literature, and he looked at a number of plasma proteomics studies in Alzheimer's disease to see what they found and how consistent they were with each other. And what he found was that the more he looked across different studies and cohorts, is that the number of proteins that overlap kept going down and down. So they really were finding very different things. So if we were just to sample, a healthy group of 18-year-olds and look that were very normal and look at their plasma, there would be a lot of variability and a lot of noise in that type of measurement. The second challenge of looking at plasma is that it has a large dynamic range of different concentrations. So the most abundant protein in the plasma is going to be albumin, and then 10 to 12 orders of magnitude lower in concentration would be molecules like interleukins or other cytokines that we think might be more interesting. So really, if we're looking for biomarkers, we believe that they're mostly going to be down here in the noise. And so as an analytical chemist, we want to have a method that will allow us to actually capture those proteins and not just only measure albumin when we get to the mass spectrometry. So we did a similar study by getting the plasma samples that were available from the Pittsburgh Center. And Dr. Mustafa Khan, who works at Takeda Pharma out on the West Coast, he was doing this work. And what he wanted to do was to first just identify which proteins were different in people that had Alzheimer's and those that didn't in plasma. And so in doing so, he took all the plasma samples that we had availability to, and he divided them into two sets. One we're going to call a training set, one is a test set. And then each one of these has people with Alzheimer's, without, and they were separated based on their race and ethnicity. And then he took them through a proteomics procedure, very similar to the one I described, except now we have an immunodepletion step. And what this does is it depletes things like albumin and up to the 14 most abundant proteins that are present in plasma. So there's a lot of sample processing that happens. And then the next questions that we had from this data set were, from the proteins that we potentially would identify that were different in people with Alzheimer's disease, could any of them be used as potential diagnostic biomarkers for disease? And if they could, did the race and ethnicity of the participant improve or impact the performance of how well that biomarker would work? And so that was work that was done with Dr. Heather Desir at Kansas, who does a lot of proteomics work, but self-taught or self-machine learning. And so she contributes a lot to these projects. So volcano plots are similar to the ones I showed before, except you'll notice there's a lot less proteins that are changing in plasma than in brain, again, due to all of that variability. But if you look at everybody, you can see that the profile of the proteins that are changing generally is largely driven by the non-Hispanic white participants in the cohort and less than the African-American Black participants. So what we did with the machine learning was we took proteins that came from everybody that were different, and then we fed them into the model. We also would try taking proteins from just this group, fed them into the model, or from this group and fed them into model. So we did a lot of different iterations to basically see which model would have the best performance. And then we used protein data in the model. And then in another instance, we would use protein data plus how old was the participant, what was their sex, what was their ApoE status, and how much education they had in terms of years. Okay. And this is an overview of how the model performed. So if we took samples from everybody and looked at how it performed from our training set and looked at how it performed in the test set, then we were about 68% accurate with being able to determine who had Alzheimer's disease, only based on just the protein data. If we added in the other variables with it, then our accuracy went up to 74%. If we started to ask the model, how well it performed on just the non-Hispanic white participants compared to the African-American Black participants. The model had better performance in just the training or just the test data set. If we added the clinical variables, then you can see that the performance would go up, but it didn't improve the performance for the African-American Black participants. And so we tested this in the training set, we tested it in the test set, and we flip-flopped it so many different ways. But overall, when we were looking at the performance, generally we could get higher performance of the model for the non-Hispanic white participant samples and not the African-American Black samples. So we really were surprised at this because even when we fed it the proteins that we knew were different in African-American and Black participants, we weren't able to get a significant performance. And so it made us question whether or not all of the clinical variables that we were using to help increase the performance were actually the best clinical variables to be using for that group as well. So years of education doesn't necessarily equate to quality of education. I said before, ApoE had mixed results in terms of its effect size, so maybe that's not the right genetic factor. So that was kind of one way that we think about looking at the biomarker work. The second way that we thought about this was to go and actually look for strong biomarker candidates from data sets that were already existing in the literature, and then compare ours up against that one. So we went and we pulled 4 different data sets that have been published using proteomics. These were looking in brain tissues. And here what you see is the amount of diversity in the data sets. And so either African-American participants, non-Hispanic white, Hispanic, and then other would mean either they only said other, or in some instances they defined other as Native American or Pacific Islanders, or potentially biracial. So we pulled those data sets. All of these had in common that they did a similar type of proteomics protocol to what I described. And we were able to isolate close to 10,000 different proteins. But we had to go in and make some decisions about which of those proteins we were going to use to train the model and to classify people that had Alzheimer's disease. So we did that, and then we tested what came out of the model up against the data set that we had generated in the first part of my talk. Okay, so here's the result that we found the first time that we did this iteration of the model. So if you want to know who has Alzheimer's disease in these participant samples, you can very accurately identify that if you measure amyloid precursor protein. That's the protein that makes the plaques. So that made a lot of sense. However, as you start to increase the number of African-American participants in the cohort, then you also recognize that there are other proteins that have good performance as well. So heat shock protein V1 also had a really good ability to discriminate who had Alzheimer's disease. So again, this just points to the fact that if you want to have some biomarker that works for everybody, you actually have to make sure you're measuring it in everybody and that it gives good performance. Or if you want to have more personalized medicine or population health approaches, then you actually have to make sure you're sampling enough of different aspects of the population to figure out what would be a better biomarker. We think we should have one that's universal. So everything that we've done mostly has been focused on proteins. So in this last bit, I'm going to share how we've looked at completely different set of molecules in these samples and kind of found some similar findings. So we've done a little bit of lipidomics work. And so naturally, work with Dr. Khan to do this work. Way more straightforward in terms of the sample prep with lipidomics than with the proteomics. So basically, you can do a chloroform methanol extraction, get your lipids out. We spike with an internal standard that has 14 different heavy isotopically labeled lipids of different classes. And then we can do some straightforward LC mass spec. All of these experiments, though, are targeted. So we actually have well-defined mass spec assays to look for each specific lipid instead of just discovering whatever we can find. So when we do this work, we start out with about 1,160 different targeted mass spec assays. We have to do some refinement of those assays, and then we can focus on specific lipid classes. And so here we decided to focus on only phospholipids and sphingomyelins, and then only those lipids that had really good reproducibility of their signal. And so these are the different types of lipid classes if you're familiar with lipid biochemistry. So a lot of lysophosphatidylcholines, ethanolamines, sphingomyelins, onidazoles. Essentially, we get very similar answers, even though we're looking at a different class of molecules. So here we have a really good ability to discriminate people that are cognitively normal from those that have Alzheimer's disease by looking at this 329 lipid profile. So that's really cool. But then we have the question of how does that clustering work if we look at the race and ethnicity of the participants. So here you can see that it's not as separated based on being cognitively normal. However, for the participants that had Alzheimer's disease, there's a clear separation of the lipid profiles that come from the African American and the non-Hispanic white participants. So I don't have answers as to why those things are different. And actually, I think it's too early to actually answer why, because I don't think we have enough sample size to make some generalizations. But what we have been able to see from lipidomics analysis is that there are some potential really good biomarker candidates for diagnosis that would work in at least these two groups that we've sampled here. So here's one example of phosphatidylserine, 18O, and this is one that has a really strong signal and it changes reproducibly and very similarly in both groups of Alzheimer's disease participants. Okay, so these are the kinds of things that we do in my lab. What I've shared with you is some of where we started and the pilot work that we've conducted. But we've learned a lot along the way, both population health focused, analytically. There's so many things we learned about this. And one of the things that we learned is that we have to be very mindful of how we conduct this research that could potentially do good, but also do harm when we bring in conversation about race. So, race is a social construct, and we have to think about what kind of misassumptions we're making in the way that we classify people and put them together. We have to think about the biases that we have and the samples that we get and where they came from and our study designs, and so many different things that we have to think about in trying to conduct these types of proteomic studies. The other thing we have to recognize is what I kind of alluded to at the beginning of my talk, which is it's not as simple as just race and having Black individuals in the study and non-Hispanic white individuals in the study. We actually have to understand the people in the study. And that means understanding not only genetic factors, but their environmental factors, social factors, all these things that can contribute to the biological signals that we see. And if you don't understand, anything about what I mean by that, if you are stressed, your biology and your plasma changes, and that can be measured. If you are discriminated against, that can actually also be measured in your biology. So it doesn't matter who you are, but there are many studies that point to those kinds of things. So the way that we're tackling this now is we have a study called PRADA, which is looking at one of the major risk factors for Alzheimer's disease, which is hypertension, and trying to understand how that actually lends to Alzheimer's disease risk in the African American population. So you all are in this area, and I'm in this area of the country, and what you notice is that there's way more red in this area, which means there's way more stroke, there's way more obesity, cardiovascular disease, and hypertension. And so we've been able to access a number of plasma samples that come from individuals in this region who have hypertension and who don't, so that we can evaluate their plasma using proteomics. And then we also have partnered with other centers across the country in the Southeast and Chicago area, New York area that had samples that came from the West Coast to try to understand what the proteome profile actually looks like in Alzheimer's disease and the heterogeneity within just the African American Black population. So we have about 4,000 plasma samples to process and close to 200 brain samples to process. So the automation and multiplexing has really kicked in. So hopefully I'll have some results to share here in the next year, because this has been a few years of work and setting up protocols. But that's kind of where we're at in driving our project. So I hope I've been able to do a lot in today's seminar, teach a bit about mass spec, teach a bit about proteomics, teach about some aspect of Alzheimer's disease maybe you never thought about, and why it's important, and how an analytical chemist can find their way. to advancing a really important problem in society. So with that, I'm happy to answer questions or have discussion, and thank you for your attention. And thank you to those on Zoom.
So,
Yeah.
so the biomarker results are, I think they're pretty good for predicting Alzheimer's. So are the results good enough for actual clinical use, or is it still more in the development?
I would say our work is in the pre-development, and there would need to be a development stage for them to get to like actual use. So in order for something to be a clinical biomarker, you have to really demonstrate and characterize so many analytical features of it. So like we really need to understand like the levels of it. The method has to be able to have very clear dynamic range, so much sensitivity, specificity, all these things the FDA would want to see. So I think we have some really good candidates from both protein and lipids from our data set that we could do more targeted work in a larger number of patient samples to just try to replicate and reproduce that those markers that we think would be good actually are good candidates. And then once we've done that work, then we can work on like the development requirements for like FDA, that kind of a thing. Thank you.
I'm curious to what extent post-translational modification influence the output of your analysis, and whether the data sets that you acquire can give you any insight into differences in post-translational modification patterns.
Yeah, so in the way that we run our experiments, we can get access to some limited number of post-translation modifications. So we can pick up things like oxidation, acetylation, methylation, things that are fairly easily to search, but also that would spray very well in the conditions that we ran. You brought up a really good point in that we are missing signals from what we've done that relate to phosphorylation, other types of modifications that might show up. And so in order to understand those, we actually would need to have some different enrichment protocols. So like phosphorylation, there's chemistries that you can use to just pull down phosphoproteins and then measure those and compare that against what we've done here. So we actually are only looking at global expression in these experiments, but I think a lot of what we see and why those changes are could be informed by a number of different PTMs. Yeah.
I'll defer to the student.
Okay.
So I apologize, I missed the proteomics part of it. The lipidomics is more targeted approach. For the proteomics, what is the biosensor that you're using? Is it fluorescence based?
So it's mass spectrometry based. So and the way that we run our experiments, it's discovery based, and that we're looking at any given point in time for the most abundant of signals that are coming off of an LC column at that given point in time. And those are the things that we send through the rest of our pipeline. We do have some new technology in the lab that uses qPCR detection and amplification, and ultimately fluorescence to do some proteomics analysis too.
I'm curious if your group has done any like reverse translation back to evaluating some animal models of Alzheimer's and see any of the same types of patterns as human samples and help us build actual good models.
Yeah, so we haven't done it in like a reverse translation type of way, but we have another body of work that we produced looking at the whole system of APPPS1 animal models. And there we've looked in like brain and heart, liver, lung, kidney, like peripheral tissues. And so the one thing that's very consistent is amyloid precursor protein. That one definitely shows up. And a lot of the things in like the glycolytic pathway, but it's not as straightforward. So there are like so many pathways in the mouse proteome that are not changing in the human proteome. And that is across different types of Alzheimer's models. So we've looked at the APPPS1, but we've compared our data sets to like APPS1 crossed with tau, and APPS1 crossed with tau and ApoE, and the 4 times transgenic. And it's just so different when you get into humans than mice. So, no.
We had a student last ask on Zoom that you'd mentioned biological differences and understand that people as a whole are not just race. Would you say that the third world countries and people in less fortunate areas would show different results, assuming they have different diets and possibly more stress in some areas?
Yes, I think genetics, environment, stress, what you eat, like all those things factor into the profiles that we would see. And I think what would actually be interesting is to be able to have access to all these different socioeconomic and lifestyle factors, and this very comprehensive data collection on like the people themselves, and then compare that with the profiles, because I think you would find that the largest driver likely would not be something like race and ethnicity. It would be like the conditions within which people are living across backgrounds. So no one has actually conducted that study yet as a collective, but there are different Alzheimer's studies in different parts of the world where I think some of that work is starting to happen. And thank you for your question.
My question is come from like how many minimum cells need to like I can see where you're
Yeah, so single cell proteomics is a thing. I don't think directly it would be applicable here just yet, but like so like for the postmortem brain tissue, we have regions of tissue that we start with that are anywhere from 20 to 75 milligrams of tissue from that region. And then that's where we're homogenized and moving forward. So I actually haven't done a count to figure out how many, cells will be in that region, that amount of tissue. When we do blood plasma, there we work with about 80 microliters of plasma from each patient. And mostly we start with that much because we're going to do those depletion steps to work with. There is the capability to do like single cell proteomics, but we don't have, we're not set up with our technology to do that in our lab.
Another question is I'm just curious about like have you checked your result available RNC?
Yeah, so not this particular study, but we do have some publications that we're a part of from a group that's looked at things like VEGF and different isoforms of VEGF. And so there's like proteomic data, CSF data, genomics data, RNA-seq data that are all pretty consistent in that way. So some of these data, some of the samples that we have, those complementary analyses were not designed into the original study design. And so then it may not be accessible to actually run those experiments. Some of the newer like cohorts and biobanks have kind of tried to think about like every kind of analysis that would be helpful to run. And so they actually have it built into their protocols. So for instances where that's available, we can go back and like pull that data or request to pull that data. Yeah, thank you.
So you talked in the early part of the presentation about how the incidence of Alzheimer's is rising over time, and you also talked about how that can be explained through the aging population. Do you think that it could also be possible that the accumulation of endocrine disruptors like PFAS and POAS chemicals could also be contributing to the development of these lesions and aggregates in the brain?
So I can't speak to those specifically, but I can say that there are a number of environmental factors that contribute to incidence of Alzheimer's disease. And so when I lived in Pittsburgh, it was very clear that there were a lot of older adults in that area, but that the incidence of Alzheimer's disease was elevated compared to like other parts outside of the city and across the state. And in part, they thought that had to do a lot with like the exposure from like steel mills and other things like that. So I do think it's some, environmental, I forget the word that they call it, but it's kind of like almost a part of exposomics and different things, like understanding the environmental conditions and exposures that people have that can lead to, plaques or oxidative stress or other things that increase risk. Yeah. Yes.
All right.
Oh, sorry, Rashni.
So, so it looks like the goal for the biomarker is a single point in time. Is there a thought of doing like a baseline and then for change in a marker over time or how?
Yeah, so our larger project, we actually have a couple of time points. So like baseline is usually really hard to get, but if you can, we can get baseline. Or like 10 years after baseline. And then you can look at like later points over time. So that's very helpful, but what's really needed is actually looking like pre-baseline. So like looking a lot, like setting back where the baseline is. So looking a lot earlier because there are people that have pathology for Alzheimer's disease that can happen 15 or 20 years prior to when they actually have the onset of symptoms. So I think it would be helpful to like, my age, younger ages, know like what that baseline is and how it's changing kind of over time in that trajectory. Because of the technology that we use, we actually have ability to do that. So with the multiplexing that we do, if we had access to samples from multiple time points, we could design it in a way that we can embed in the longitudinal aspect. It's possible in terms of the data analysis. It's not straightforward. Yeah, it's not straightforward to add another, add a whole nother variable, but it's possible.
These are like super nerdy questions, but you talked about trying to look at those really low abundance compounds. So do you just do reverse phase, or do you do like reverse phase antihelix to increase overall protein coverage?
Yeah, so first for plasma, we have the immunodepletion that we do. And then when we have our peptide mixtures after labeling, then we'll do high pH reverse phase chromatography. And so there we collect every minute for about 90 minutes. And then depending on how much instrument time we have available in the depth, we'll condense those down into a smaller number of fractions. So for these experiments, or for the most recent experiments, that would be like 24 fractions is kind of what we're doing now. Yeah, and then each one of those fractions gets low pH online.
For that second low pH like run, or
We've just done like self-packed capillary columns that we don't have in that. Yeah.
then acquisition wise, like I saw you're doing like MS3 in some of the cases. Are you guys doing like DIA or like what's like the sort of like acquisition side of it?
Yeah, so we do data dependent acquisition, so which means we're taking the most intense signal that comes out and then we're breaking that peptide apart, and then we'll do that up to like 15, or we'll do as many as we can within 3 seconds, depending on which mass spec we're using. So that's kind of the cycle that we are using.
And is there anyone doing Alzheimer's stuff top down side, like results correlate, or is it pretty much you kind of have the space from bottom up?
So there's several of us that have this space from bottom up. And so there are a number of groups that are doing different things in the space. I wouldn't say necessarily disparities, but just in general. Top down, a lot of what people are interested in isn't like fully discovery work. It's usually like focused on specific type of molecules. So there are a couple of folks who are really interested in like apolipoproteins. And so they've designed some methods to like pull down all the different isoforms of apolipoproteins and look at how they're changing and things like that.
Thank you.
Yeah, it's easy if it's one protein top down. Yeah.
Do you know whether common medications that might be used by either your control groups or your treatment groups might differ between your subgroups, and would they differentially affect them?
Yes, I think there's reason to believe that they would affect the proteins. However, in the way that we matched, we didn't match based on what people were taking because we just had so few in our African American group, but we have access to figure out what medications they were taking. Here, that's not as much of a focus, but like for the last study I described, where we're looking at hypertension, that one we know a lot about their medications, and so like we've excluded certain participants from being eligible to be in the study if they have like some other major chronic conditions that would impact the medicines that they're taking. And then we know what types of like antihypertensive medications our participants would be taking. So we look for things like that. We didn't design the study in a way to answer the question though about like, the impact of the different medications. So I don't know if we're powered to do that, but yeah. So I believe it could be possible to like redesign a study to do that. There's so many things, like the medicines, the diet, which we don't really know.
Yeah.
Other things. Yeah, the medications and also the incidence of the comorbidities too. So we do report out like in our papers what percentage had, hypertension, obesity, like other things too. But yeah.
Good morning. You mentioned in clinical testing how there are some higher invasive and diagnosis that aren't for everyone, or so is it also possible are overdiagnosed Alzheimer's or currently you have verbal conversation with the doctor and look at cognitive difficulties and just automatically assume, oh, it must be Alzheimer's.
I actually think it will be more under diagnosing than over diagnosing. More so because not there are parts of the population that aren't informed or are aware that dementia and Alzheimer's disease are not normal parts of aging. So there's some individuals who actually may not go and see about it. And so I think they would go undiagnosed for like some of those reasons. There are some researchers that are doing work to make sure that like the cognitive tests and batteries and things that are conducted on patients are culturally tailored and don't impact diagnoses. So there's like those kinds of things that happen that are also happening where like maybe the way that the question is asked would lead to an improper diagnosis on anybody. So trying to figure out how to tailor those things according to the different demographics that they're interested in. But yeah, thank you. Y'all got some good questions.
The RNA-seq. Have you considered looking at geospatial transcriptomics for single cell RNA-seq? In other words, you'd be able to take your tissue and actually be able to see its labeled where each of these proteins are setting up. That part.
Got it. So we haven't considered doing the geospatial transcriptomics in this project. But we do have another R01 that's in collaboration with Jeff Spraggins, who is an imaging mass spec person at VUMC, and Matthew Schrag, who's a neuropathologist at VUMC. And in that work, we're using multimodal imaging technologies to map Alzheimer's disease. So what that means is we can take a slice of a brain tissue and know the coordinates of where that slice is coming from in the entire brain region. There's pathological information and staining that comes from some part of the team. There's also the chemical imaging mass spec where you can look at mostly you're seeing like small molecules, lipid profiles, that kind of thing. There's also like fluorescent imaging pieces that are incorporated into it. We get a portion of the slice, and then we do like homogeneous proteomics based work. There's another group doing a different type of proteomics, and there's probably some stuff I'm missing. That's part of the project. We're just the proteomics arm. But in that way, like there's a really big look across different types of modalities to understand very, a lot of complex information. And then there's also the patient information that can be mapped onto a specific, specific regions. So it's a work in progress. We're about like 2 years into that project. I'm doing too much. I think I'm doing too much. Thank you all.